Lauren Schmitt, who's in the Perfecto Lab here at University of Michigan, and she's here to talk about the effects of species and location on litter decomposition of coffee.
Thank you. So as he said, my name is Lauren Schmitt, and I'm a third-year PhD student in the Perfecto Lab in the School for Environment and Sustainability. And today what I want to share with you is a little bit of the research that's going on in my lab group, and then go a little bit more in depth about one of the projects that I've done on decomposition in coffee agroecosystems. So many of us likely started our day with a cup of coffee. Millions of cups of coffee are consumed every day, and there's a growing awareness of where this coffee is coming from. So coffee is grown globally in tropical regions, often mountainous tropical regions. And there's a wide variety of what these coffee farms and coffee agroecosystems look like. And so all three of these pictures were taken in the Soconusco region of Chiapas, Mexico, which is the area where I work. And you can see this picture here in the upper left is an area that was recently cleared from forests to convert into coffee farming. And a few trees remain up here on the top of the ridge, but it's certainly dominated by these coffee plants here. This picture down here, it's a little blurry, but the main feature, there's a little more vegetative complexity here and more trees. But again, it's these rows of coffee up and down the mountainside that is the main feature here. In contrast, the picture on the right, at first glance, you might not realize that it's a coffee farm at all, though that is indeed what you're looking at. This is the system that I work in and many of my lab mates as well. So this is sort of what you're seeing here is coffee plants, sort of shrubby at maximum about 1 meter high, growing in the understory of these larger canopy trees, and then there's an herbaceous layer beneath this. And so we like to think about this, as Jennifer was just saying, as an ecological system in which agriculture sort of just happens to be taking place. And so a lot of work has come out of my lab on a variety of ecosystem services and functions in this system. Some of my lab mates are focusing on the herbaceous layer and the flowering resources that they provide, on the bees and the pollinators, the hummingbirds, and their role in the ecosystem, potentially as biocontrol. A lot of work has been done on the ant communities. There's a keystone ant species in this system, and their community dynamics and trait-mediated interactions. On bats, on landscape level effects, on fungal diseases, on parasitoids, the list goes on and on. But what I'm focused on in particular is the plants themselves and what happens to those plants when the leaves are senescing and when those plants die and when it becomes part of the detritus and part of the leaf litter. And so the framework that I sort of like to orient myself around starts with, in the center here, a coffee plant, which of course has all of its physical characteristics, but also its chemistry. And we hopefully in this room all appreciate how interesting plant chemistry can be. When speaking about coffee, the main sort of chemical of interest, both as a coffee drinker and an ecologist, is caffeine. And so caffeine is a secondary defense compound, and so a deterrent to herbivores. And so it's interacting with these upper trophic levels here, but it's also still present in that plant material when sort of nutrient cycling is happening and during decomposition. And of course, there are both direct and indirect interactions taking place between all three of these levels, though what I'd like to focus on today is this arrow here and the decomposition of coffee leaf litter. And so there's already a good amount that we know about the decomposition of secondary defense compounds. They have been found to alter detritivore and decomposer communities and inhibit nutrient cycling. And if you think about it, this makes good sense. If secondary defense compounds are a deterrent to herbivores, they might also be a deterrent to detritivores and decomposers. But on the other hand, caffeine is an alkaloid, and by virtue of being an alkaloid, it has nitrogen in it. So you might also think that in a nitrogen-limited system, there is some extra incentive to decompose this leaf litter that has caffeine and thus nitrogen in it. And so regardless of which pathway it is that is dominating here, we know that time and space matter quite a bit. And so by space, I mean, where is this leaf litter decomposing? Is there any home field advantage? The idea that there might be an acceleration of these processes near the source of that organic material. And then we also know that time matters a great deal. And so these graphs on the right here are just meant to show sort of the alkaloid concentration over time between a species of fescue with and without the alkaloid. And you can see that the alkaloids are gone from this leaf matter relatively quickly in the decomposition process. So if we want to look at their impacts, we need to consider whether we're looking at sort of the very beginning of this process or the longer term effects. And so I studied this, or I asked this question in a coffee system for the reasons that I've already mentioned, the amount of land dedicated to coffee and its importance, but also because there are two main cultivated species and they differ in their caffeine concentration. So it's an appropriate place to ask this question. So the two main cultivated species are Coffea arabica and Coffea robusta. Arabica is about 3/4 of global production. It has a higher market value. It is generally grown at a higher elevation and has a lower caffeine content. Robusta is about the other 1/4 of global production. It can be grown all the way down to sea level. It has a lower market value, but it's a hardier species, in part due to its higher caffeine concentration. And so sort of the research that has been done that's looked at the effect of caffeine in these agroecosystems has mostly looked at its impact on herbivores and on herbivory. And there has been some sort of deterrence to herbivory shown, particularly amongst generalist herbivores. Specialists often have some sort of mechanism to deal with the caffeine. And so sort of with this background, the question that I wanted to ask is, what is the effect of species, comparing Arabica and Robusta, and the effect of location on the decomposition of coffee leaf litter? And so I hypothesized here that Arabica would decompose more rapidly than Robusta, in part due to its physical characteristics. So Arabica leaves are smaller and thinner than Robusta leaves, but also because of that lower caffeine concentration. I also hypothesized that decomposition in a forested control site where coffee is not usually cultivated would be slower than in areas where coffee is grown. And I thought that this would be the case if there were distinct decomposer or detritivore communities that are not sort of used to decomposing caffeine and coffee leaves, or if the composition of detritus in these systems were different. So it's not just coffee leaf litter. In the areas where coffee is grown, you saw earlier that there is that overstory, but that's going to be a smaller number of species that are managed and chosen by the farmers. And so I did my field work here in Finca Irlanda, which is a 300-hectare shade organic coffee farm right on the border of Guatemala in Chiapas, Mexico. And it's situated at an altitude where the majority of the coffee grown is Arabica, but there are patches of Robusta in addition to some cacao and some cardamom as well. And so the study design that I used was a tethered line design. An aerial view is up here in the right-hand corner. And so what I did was I collected recently senesced leaves from Robusta and Arabica plants, dried them, weighed them, and tied them in bunches onto fishing line to create these lines or these spokes, and then arranged 6 of these lines into a wheel that I set out in plots where Arabica is grown, in plots where Robusta is grown, and in forested control sites. And then I would return to each site every week, collect one of these lines or spokes, dry it, weigh it, and then I used mass loss as a proxy for decomposition. So it doesn't photograph particularly well, but this picture in the bottom corner here, what you're looking at is 4 bunches of leaves tied onto a fishing line here, 2 of which are Arabica and 2 of which are Robusta. And so this took place over 6 weeks in the summer of 2016. And so these are the results of the mass loss. And to sort of orient you here, the y-axis is the proportion of mass loss, and the x-axis is the number of weeks in the field. And in each graph, the pink line is Arabica leaves, and the blue line is Robusta leaves. And so starting here on the left, in plots where Arabica was grown, so these are split up by location here, you can see that there's a significant difference between the leaf species, and that Arabica, this pink line, is decomposing or losing mass significantly quicker and more than the Robusta leaves in these same sites. There were no differences between the species in the Robusta plots or in the forested control sites, but what is important, I think, to note here is the difference on the y-axis. So both species are decomposing or losing mass slower in the forested plots than they were in the areas where coffee is grown. And so if these are all put on one graph, it looks like this. And so again, I think the important piece here is that we're seeing potentially some home field advantage or acceleration of the decomposition or mass loss of the Arabica leaves in the plots where Arabica is grown. And both species in the forested control sites are lower than they are in the areas where coffee is grown. And so going back to my hypotheses, I had hypothesized that Arabica would decompose more rapidly than Robusta. And I found this to be the case, but only in areas where Arabica is grown. And so I can't really say why this might be, if this is due to the chemical differences, due to the physical differences, or if it's due to some sort of home field advantage or effect of the location. My second hypothesis, that decomposition in the forest would be slower than in areas where coffee is grown, was also supported. But again, it sort of frustratingly, I can't say why this might be, if this is due to a distinct decomposer detritivore community or the composition of detritus. So the results that I've just presented to you here were part of some pilot work that I did in my first field season. And since then, I've been following up to try to sort of tease apart the how, the why, the when, and the where of those differences that I found in the first study. And so currently in the field, I have litter bags with these same treatments that are testing if these patterns hold up over a longer period of time. So I mentioned at the beginning that time scale is going to be very important here. And the initial study was just looking at that very first part of decomposition over the first 6 weeks. And I'm currently collecting data over a 12-month period. I've also begun to look into the effect of the composition of detritus and leaf litter. So using a similarly designed study and factorial combinations of different overstory trees to see if there are additive or synergistic effects of coffee decomposing in combination with other species. And so hopefully that will sort of tease apart why the decomposition in the forest was slower. This picture up here is at the Matai Botanical Gardens here. I have some coffee seedlings, trying to control a few of these factors that cannot be controlled in the field, and looking at allocation of nitrogen either to defense or to growth under different conditions. And in the future, I also hope to characterize the decomposer communities across these sites and really sort of parse apart the effect of the physical differences and the chemical differences of these species. So with that, I'd like to thank my lab group, everyone who has helped me with the fieldwork part, and all of my funders, and I'm happy to take questions.
Great talk, Lauren. If Dushan Sun could come down and set up your slides, that'd be great. And questions for Lauren?
It's a very nice talk. I have kind of a naive question. So Robusta has higher caffeine levels, yes? So I would sort of naively expect that in the Robusta plots, you'd have an advantage for Robusta degradation because the decomposers would be more used to seeing those high caffeine levels. Mm-hmm. But that's not what you saw, and I wonder if you could speculate maybe on why that is.
Yeah, so I also had that expectation that I might see an acceleration of both species in the Robusta plots if there's sort of usually a higher concentration of caffeine there. And what I think that might be is that the differences in the chemical composition or the caffeine concentrations are being overshadowed by the physical differences in the leaves. And so just sort of qualitatively, if you're holding an Arabica leaf and a Robusta leaf, the Robusta leaves are much thicker and tougher. And so it may be that sort of the physical constraints of processing that leaf litter overweighs or overshadows the differences in the chemistry. That would be my guess.
Yeah. Thank you.
The slope was pretty much the same, but like after week 1, they had totally different points. Like, do you know why they were kind of so different at week 1? Was there just big differences in that first week? Or?
Yeah, so that's an important distinction that some of the most obvious differences on those mass loss graphs are in magnitude, but usually when we're thinking of decomposition, we think of it as the slope, as as a rate, so the slope of that line. And so the trend lines that are on those graphs are probably are an approximation of that decomposition, but not exactly that, because we wouldn't necessarily expect that to look linear either, and that's sort of a best fit for that. But I think that that is an effect of the short time scale in which I'm looking at it. I also have subsamples of this, which I've done some carbon to nitrogen analysis, which I didn't present here. But there, that is showing that they were all sort of starting from this homogeneous part of the leaf, and so there is sort of a week 0 that is coming from one place that maybe would be helpful to add to that graph. Yeah.
Great, thank you, Lauren. Good talk.